Welcome, everyone, to our monthly Science of Alt Protein seminar. My name is Amy Huang, and I'm the Associate Director of Scientific Ecosystems at the Good Food Institute. For those of you who are here for the very first time, welcome to our community. GFI is a nonprofit think tank. We are 100% driven by philanthropy, and our singular goal is to build a world where alternative proteins are no longer alternative. So if you're looking to take a deeper dive into the science of plant-based fermentation or cultivated meat technologies, you are very much in the right place. We recommend that you check out our open access resources at gfi.org/science. So with that, it's my pleasure to introduce our fantastic speaker today, Dr. Leonie Jahn, who is focused on unlocking the potential of microorganisms to create more sustainable foods for the future. She really draws inspiration from nature and traditional fermentation practices, seeking to uncover new organisms that expand the biodiversity of microbes that are used in food production, and to develop innovative fermented foods with the potential to replace animal products. Right now, she serves as a tenure-track researcher at DTU Biosustain and acts as the deputy head of the Microbial Food section. She holds a PhD from DTU and MIT, and additionally, she's a co-founder and scientific advisor at Matter Foods, a Copenhagen-based company specializing in meat alternatives derived from fungal fermentation. I also want to share that Dr. Jahn is a 2021 GFI grantee and recently was awarded the very prestigious Niels Foss Talent Prize. This prize is awarded to talented and promising upcoming scientists who've made really significant contributions in the research domain to the application of analytical technology that improve the sustainable use of agricultural resources and help ensure food quality and safety. So Leonie, we really want to congratulate you on this well-deserved recognition. During today's talk, we're really excited to get to learn from you and really want to thank you again for being with us today. So with that, I will turn the floor over to you.
Wow, thank you so much for this gracious introduction, those kind and warm words, and also for the opportunity to speak here today at the GFI seminar series. I'm really honored and very happy to see so many people here. So thank you very much. And I will share my screen now so we can get started. So as was introduced, I will talk today about the biodiversity and the green transition from discovery to application and share my research with all of you. I'm co-PI and the leader of the Microbial Foods lab here at the Technical University in Denmark. And in the lab, we like to focus on diversity, where we actually try to map and characterize microbial biodiversity for the development of more sustainable foods, and also to expand the diversity in food fermentation processes and general food production processes in the food system. We're also very interested in discovery, especially understanding the fundamental ecological and genetic underpinnings to flavor and texture. And finally, we are thinking a lot about design, actually on many different levels, so ranging from the systems level, where we do absolute sustainability modeling, down to the cellular level, where we do genetic engineering for new functions and ways to actually improve microorganisms even further. And this talk today, I'll take you on a journey on microbial biodiversity for food innovation, ranging from discovery to applications. But first, I would actually like to step back a moment and provide you some background about why I'm so passionate about microorganisms and improving the food system, in general. And I would like to start with this image from the New York Times here. Because regardless if you study ecosystems in a fermented food or the planet as a whole, it is actually really important to recognize one fundamental principle, and that is that life and the environment are interconnected and form one basically geophysiological unit. And that means that the evolution of life and the evolution of the environment are actually intertwined and influence each other. And to provide you with an example to illustrate that better, you can imagine the world some billion years ago when it was like the atmosphere was devoid of oxygen. But then cyanobacteria evolved and later plants that were able to convert carbon dioxide into oxygen. And they completely changed the environment because over time oxygen built up in the atmosphere. And that had a huge impact again on the life on Earth, because it meant that a lot of species that had evolved on anaerobic conditions so far were no longer able to grow, or population sizes declined a lot. But on the other hand, it also meant a spark in evolution of completely new species that actually depended on oxygen in the atmosphere. So you can see that one product that initially accumulated as a waste product then sparked the evolution of a whole new branch in life. And I think that this is really important to keep in mind, especially if we're thinking of our own impacts as a species, especially in light of climate change. And if we talk about sustainability, we actually mean to keep an equilibrium in like the balance between life and the environment, or that we try to keep conditions in the environment that actually support our own life and the life of future generations of humans, as well as all the life forms that we depend on. And this is somewhat tricky because it is always evolving, as I said, in response to each other. And also we have a huge impact at the moment where we're kind of shifting towards or outside of our comfort zone. So it is really important to think about how can we keep Earth in an equilibrium, or at least in a range of environmental conditions that allow future generations of humans to continue to live here. And if we think about the question, are we there today? Are we actually sustainable? This is really important to think about, and it's really difficult to assess. And in 2009, the Stockholm Resilience Center actually put forward the framework of planetary boundaries, which are meant to assess how well we actually do in terms of sustainability and keeping this equilibrium. And you can see in 2009, 7 different boundaries have been assessed. And in the green area, you can basically see the so-called safe operating space. So within those areas, if we are kind of operating within that space, we would keep conditions that also allows future generations of humans and other life forms to continue to live on this planet. But if we are going outside of the safe operating space, we will have a huge impact on the environment, which might also cause conditions that are no longer favorable for us to live in. And you can see over time, there have been more boundaries assessed, but also more of them have been transgressed. And in 2023, out of 9 boundaries, 6 have already been crossed, which is quite alarming. So long story short, basically to answer my question, are we sustainable? I have to say no. But that also means that if we really want to continue living on this planet, we really have to get going and do changes or change something in order to also have future generations being able to continue to live on this planet. And then I actually sat down and drew a little bit with my children at home. They helped me here. Because I thought I'm coming back to this relationship between life and the environment. And basically you can see this net of different species that all depend on each other and eat each other, basically. So we have food webs. And then they also interact with the environment. And they mainly do that through eating and breathing and their metabolic activities, and then the waste products that they are producing. So it's actually not surprising that then food and our food systems also have a huge impact on the environment, as food is also really fundamental and crucial to every living being on this planet. And the Eat Lancet Commission actually published this article. And in this article, they say that food is the single strongest lever to optimize human health and environmental sustainability on Earth. So food has indeed a huge potential to address a lot of the sustainability issues that we are facing today. And there was another study published in Science where they actually said that even if we had stopped all fossil fuel emissions already in 2020, we would not have been able to reach our climate goals unless we would have also changed our food system. And that, I think, highlights again how important the food system is if we talk about sustainability. So we kind of wanted to dive a bit deeper into that, and we started doing an absolute sustainability assessment where we integrated life cycle assessment into the framework of planetary boundaries. And then we also looked at the impact of the food system or the contribution of the food system for the whole and the whole, basically on the whole impact, global impact. And we can see that some of the impact categories like biochemical flows, land occupation, and biosphere integrity actually almost exclusively derive from the food system. And also in other categories like climate change and freshwater use, you can see that the food system has a major impact on global sustainability or unsustainability, I guess. So what is wrong with our food system? So there is a lot of different things that are challenging in our food system, and I don't have the time to go through all of them, but I will start with two points that are linked to the inefficiency of the food system. And one aspect is animal farming, and the other aspect is the food system's design, and in particular its linearity. And to illustrate those points more, I have some slides here. So basically, the reason why I think that animal farming is an inefficiency is derived here from this graph, where you can see the calories that are provided by different plants that go into feed of animals. And then in the end, you can see the calories that are being consumed by humans from those animal products. And you can see that basically up to 97% of the calories are lost across this path. And I think that really highlights that all the resources that go into growing, for example, corn or soybean or other animal feed is basically lost if we rely on animal products, because yeah, we could in the first place eat those plant materials and have a higher, more calories from the same inputs, basically. On top of that, animals also produce CO2, especially cattle and cows, and when they digest plant materials, they produce methane, which is a very active carbon, which actively contributes to climate change. And manure also contributes to CO2 production. Besides the inefficiency, I think it's a personal matter to me to also highlight that basically, yeah, we have a choice at this point, at least in many parts of the world. We can choose what we eat in order to get our nutrients. And I think we can choose to eat plant-based, not only for the sake of the environment, but also for the sake of the animals and animal welfare, because I do believe that especially mammals have very similar emotions to what we experience and feel. And I think it's important to protect them and to treat them with respect. In addition, there's also public health concerns associated with animal farming, especially in intense animal farming, where antibiotic resistance evolution can take place, and then in zoonotic diseases or other transfer events, either those resistance genes or also those zoonotic diseases can actually spread to humans. But coming back to the challenges and the inefficiency of the food system, the other aspect that I mentioned is the linear design of the food system, where the food system is designed in such a way that we have input materials and then the foods as output materials, but on top of those outputs, we also have waste, food waste, and also byproducts that are currently underutilized. And 1/3 of all the food that is produced in the world is wasted, which means all the resources that went into producing them are also wasted and not utilized. And that doesn't even account for a lot of side streams and byproducts that are not even considered food waste in the first place. And I think if we then consider how to solve those issues, I think it's really wise to draw inspiration from nature. And if we look at natural ecosystems, we can actually see that none of them are linear, but all of them are circular. And that means that that these systems minimize waste, and dead plants and animals are actually decomposed, and thereby their nutrients are made available again for new life to be formed. And interestingly, a lot of in natural ecosystems, filamentous fungi play important roles as decomposers, as they possess the unique ability to break down various materials. And I think this is really impressive. Like they can break down, for example, lignin, which is one of the most stable organic molecules on Earth, and they can even break down rocks, solid rocks, and make phosphorus available to life. So they're really potent in growing on all sorts of different environments and nutrient sources. And on top of that, fungi can also be used for food production. So if you think of fungi as food, you might imagine like white bottom mushrooms, but the realm of fungi extends far beyond. So you can also think of products like soy sauce or blue cheese or beer or wine or bread, which all owe their interesting flavors fungal fermentations. And on top of that, filamentous fungi can grow in filaments, as the name says, and those filaments can resemble the fibers found in meat, so they can also create interesting textures in 3D space. So I do think that fungi might actually hold the potential to address both of these problems that I presented, so we can actually create more circular systems and also create interesting products that could replace animal-derived products. And we wanted to actually fact check our assumption that microbial foods would actually indeed be a contribution to the sustainability. And thereby we modeled basically the impact in an absolute sustainability framework, the impact of our current business as usual diet, which you see in deep red or purple. Then you can see in yellow the diet or the impact of the Eat Lancet recommended diet, which foresees a lot of changes to our diets, which should make it more sustainable. And then we also tested 2 different diets where we replace all the red meat from the business as usual diet, but otherwise leave it as it is. And we replace it by something we call microbial food. And we have 2 different types of microbial food that we are modeling here. One is a food where we ferment a byproduct from food production with a filamentous fungi and replace the meat based on a protein, on a 1-to-1 protein basis. So it's derived from solid state fermentation. And then we have a microbial food that is derived also from a side stream of food production from corn or wheat stover that is then saccharified into sugars and then used in liquid fermentation to grow microbial biomass. And what we can see here is basically that the microbial foods performs in most instances equally well or better than the Eat Lancet recommended diet, which is quite exciting, which means, and of course better than the business as usual diet, which means that we can indeed have a huge contribution when we change our diets, and that microbial foods also have a huge potential to improve the sustainability of the food system. However, we should also notice that we are not able to go into the safe operating space by changing to those diets for most of the categories, which highlights that we need multiple different changes in the farming and food system in order to actually make it truly sustainable. And another observation is that the liquid fermentation actually comes with some trade-offs. So you can see that, for example, in the freshwater use, it's performing much worse than the business as usual diet and the other diets, which highlights that solid state fermentation is likely the best way to go forward. Inspired by the potential of filamentous fungi for food innovation, I co-founded a company that is called MetaFoods, where we use 5 whole food ingredients, namely split peas and lupin beans, beetroots, potatoes, and oats. And through fungal fermentation, we can convert those ingredients into delicious meat alternatives. And we have already some initial products on the Danish market. And of course, this is a team effort. And here you can see our amazing CEO, Randi Wallsten, cooking up some samples. And of course, if I'm saying we and our efforts, I mean especially her efforts as well as the whole team at MetaFoods. And also at the university, we've been exploring the potential of filamentous fungi for food innovation and especially for circular systems, looking at waste streams. So here you can see Nabila Rodriguez Valeron, who holds a PhD from Harvard and the Basque Culinary Center, and also worked at the 2 Michelin star restaurant The Alchemist here in Copenhagen, at the site while doing her PhD. And here you can see her making some misos. And misos are traditional Asian fermented foods where you use a starter culture, a filamentous fungi called either koji or Aspergillus oryzae, and then growing it in a koji, which is kind of a pre-culture, usually grown on rice, and then you inoculate soybeans with that pre-culture and ferment it over several months and sometimes even years to form a miso. And here you can see Nabila making some misos based on side streams. So she used wheat bran to grow the koji and then brew a spent grain to make the miso. And then she did some consumer tests actually, where she used the miso as a condiment, as it's usually done, to improve the flavor properties of plant-based foods. And she even tested it on endive, which is known to be really, really bitter. And basically the bitterness was, it could mask some of the bitterness and increase the liking of a lot of plant-based products, including the endive, which is quite promising because that means that we can use a waste stream through fungal fermentation and then improve the liking of plant-based diets. We further screened a lot of byproducts from the food industry for food fermentation. And we developed a mycelium puff pastry, which is now further developed at the Alchemist restaurant. And I do think this is maybe not exactly what will save us from climate change and be served to a lot of people. But at the same time, I think it really illustrates the huge potential in terms of what we can do with fungi and what we can envision them to do and all the different textures and basically applications that we can do if we innovate with filamentous fungi. We also developed a food product that has a flavor that is really, really similar to chicken, which is really exciting, and we're continuing this work at the moment with further follow-up experiments and optimization steps. And we also developed a fish finger alternative that has a rather neutral flavor, but a really interesting texture, very similar to actual fish fingers. So there is a lot one can do, and for all of these projects, we used established filamentous fungi that are traditionally used for food fermentations. So there's huge potential for food innovation here. And for all this work, where I said we, I actually mean that it's Kim Weindorp and Luus van Dam, who have been doing all this scientific and culinary work together with some collaborators, but they are the driving force here. But then if we think about food fermentation and the molds that are traditionally used for food fermentation, it's actually very few species that are utilized around the world. And I thought it would be interesting to see, because there's millions of species of fungi known, if we could actually go beyond those known molds. But on the other hand, fungi are known to produce secondary metabolites and bioactive compounds that can sometimes be toxic. So there's mycotoxins that can have actually huge health impact. So it's also a question of like where to start and how to do this safely. So I thought, okay, there's several thousand species of basidiomycetes, which are mushroom or fruiting body forming fungi, and several thousand of those fungi are known to be edible. So we thought it could be an interesting starting point to look at the mycelium of the edible mushroom forming fungi to see if we can innovate with those. But if you think of a potato plant, for example, you can eat the potatoes, but you cannot eat the fruit of the potato plant because that is poisonous. So we thought we better taste this, not taste, sorry, test this before we go ahead. And interestingly enough, we were in touch with The Alchemist with other ongoing work. And they had developed a dish that they call literally the Petri dish, which is a Petri dish, where they developed a edible agar, and then they grew the mycelium of the oyster mushroom Pleurotus austriatus on it. And then they are adding some delicious topping to it before they're serving it in the restaurant. And maybe I should say that, of course, they are pursuing a lot of deliciousness in their dishes, but they also like to play with emotions, and this dish maybe also plays a little bit into that. Yes, but we thought it's a really interesting test bed to test our hypothesis, or in general to test the feasibility of using mycelium for food innovation, and especially establish the safety. So what we did was, well, here you can see, first of all, the fruiting body, the oyster mushroom, and again, the mycelium. And then we compared the growth. Oh no, now it's low resolution again. I had just changed that, but well, never mind. But basically what the graph is supposed to tell you is that basically it's faster to grow and harvest the mycelium than to harvest the fruiting bodies, which is of course beneficial from a sustainability point of view. And we also grew the mycelium on a number of substrates and different carbon sources, and could identify that it would grow on a very broad range of substrates, which are also often present in different waste streams. So there's also huge potential to use it on different waste streams. We then also did a pangenome analysis of our strain compared with other strains of the same species and genus. And there's some similarities, but also some differences. So we looked a bit deeper into that. And we can see that there is an open pangenome, which means that if we sequence more strains, we're likely to find new genes that would be added to the species. And we can also see that there is a core genome, but that is really small with only 754 genes. And then there's a huge accessory and unique genome, which means that a lot of the strains have very special features, one could say. And then we looked a little bit deeper into those special features and where they group and which kind of parts of the functionality of the fungus. And interestingly, like all of the accessory and unique genes can be found in secondary metabolite production, which relate both to the safety of the mycelium, but also to the flavor production. So that means that if we're testing different strains of the same species, we might be able to actually also find interesting differences for the culinary potential. But as I said, some fungi are known to produce mycotoxins. So we did metabolomics to actually test for the presence of any known and regulated mycotoxin. And we didn't identify any mycotoxins in the fruiting bodies nor in the mycelium. But then oyster mushrooms are actually known to produce also protein-based toxins that are heat sensitive. So if you cook the oyster mushrooms, they are safe to eat afterwards. But we still wanted to double check how this performs, or if the same mycotoxins are produced in the mycelium. And then we checked and found all the genes for that also in our strain, and grew the strain both as mycelium and fruiting body, and compared the expression of those genes. And we can see that the expression is much higher in the fruiting bodies. So we are concluding that if anything, the mycelium is actually safer to eat than the fruiting bodies. We also looked into other aspects of the culinary potential and consumer acceptance and willingness to eat. And we're concluding basically that the mycelium of oyster mushrooms is actually a very promising source of alternative protein, and has a lot of gastronomic potential, and could be and should be explored further. And then we thought, okay, this is really interesting. Maybe that holds true also for other basidiomycetes. So we actually built a strain collection of more than 100 potentially edible strains, which are either molds used in traditional food fermentation or fungi with edible fruiting bodies, and tried to cover a broad phylogenetic diversity. And then we measured the texture of all of those strains compared to different meats and seafoods, and also other foodstuffs. And we could actually identify interesting strains that have interesting textures that get very close to meat and seafood, and like tofu and other food products that are quite appealing. And we're currently following up on those findings and see what we can do with them. But as I said before, there's a lot of biodiversity, and even if we are counting all the species that are found in traditional fermented foods and starter cultures and even the edible mushrooms that we have, we're still only looking at this yellow thin line on this plot here. And then there is like this similar, also quite thin line in purple of all the characterized microbial species. And for example, the company corn, or the product corn, was developed of a fungus that produces actually a mycotoxin, but only under certain conditions, and they can produce it safely. But it's a strain that has previously not been used for food production, but was characterized. So also in that space, there's innovation possible. But then there's also this huge blue space with the estimated biodiversity of bacteria and fungi, which means there's a lot to be done still to actually find new interesting species with a lot of potential for culinary applications and for developing sustainable and delicious foods in future. And I also want to say that this study was again led by Luce van Dam, and here you can see her at the Alchemist restaurant together with the former R&D chef there, Diego Prado. They're testing some fungal samples there. And to summarize this section, I would just like to say that fungi, and especially fungal biodiversity, are a true treasure trove for the development of delicious plant-based foods in a circular food system. But then I think it is also important to consider that, to think about basically all those flavors that are produced by fungi and why they are produced. So basically, some compounds like umami and free sugars arise during the fungal digestion, which is happening outside of their bodies when long fibers and proteins break down into their individual building blocks that we can then perceive as umami or sweet. Other compounds like organic acids are results from the fungal metabolism and end products of the metabolism, and we can perceive them as sour. But then there are all those volatiles and interesting, delicious, and delicate aromas. And some of them have actually been shown to be produced by the fungus as communication molecules and communicating with itself and also other species of fungi, but also bacteria. And I think just to highlight the impact of that, if you think of truffles, the truffle flavors are actually not produced by the fungus, but only in the interaction of fungus and bacteria when they are talking to each other. So yeah, I think that just highlights the potential of these relationships and the importance for flavor. To study that in a bit more detail and to look at the effect of microbial interactions on flavor, we started a small research project at a university where we assessed the relationship between two commonly used species for food production, Aspergillus oryzae, the koji mold that I mentioned before, and Bacillus subtilis natto, that is used to produce natto in Japan, which is a soybean-based ferment. And we just put them together and on a plate, and you can see that the Aspergillus grows in very circular colonies, but then the Bacillus grows more in an oval shape and tries to escape the fungus. So there is something going on between the two. And we wanted to also look at that from a food perspective. So we made a natto basically of barley of the Bacillus subtilis, and we made a koji also of barley by fermenting it with the Aspergillus oryzae. And we also made two products where we mixed Bacillus subtilis and Aspergillus oryzae. And then we wanted to ask people about the flavor, but we also thought, okay, the texture and appearance is quite different. So we made a cryo extraction, which is preserving all the flavors, but comes in a liquid, so we're not distracted by any flavor, by any textures or appearances. And then when we did the sensory analysis, we could actually see quite some differences between the three different groups. And what I think is really interesting and exciting, that basically the mixed culture was liked the most and had the least strange flavors and aromas, as people reported. Which actually highlights that the diversity within a fermented food is it's basically beneficial. So it's before we talked about basically the biodiversity of filamentous fungi and how we can innovate it with individual species, but now we have a community, or at least several species, and a more diverse setup, and we can see that that is also really interesting. So it's not only about expanding the biodiversity of individual strains that are used for food production, but also within foods that are fermented, diversity seems to be beneficial. And the people who have led this work are Carola Hesemann-Rosenkilde and Tiffany Mack, whom you can see here on those pictures. And I think I just summarized it already a little bit, so I will move forward in the interest of time. And because we got really interested in the diversity and microbial communities that can actually also shape flavors and textures, we thought we really want to investigate which factors then have an influence on communities that are formed. And then we started by looking at substrates and starter cultures. And this is, again, work that has been done by Nabila. And she made, again, kojis and then misos on 2 different substrates, sorghum and barley. And then she also used 2 different starter cultures of Aspergillus oryzae with a slightly different genetic makeup, and then made some misos out of that. And what she found was that the strain has a small effect on some flavor compounds, for example, curcumi peptides, but the substrate overall is actually the major driver if it comes to differences in the flavor landscape. A similar conclusion was also reached by our collaborator Joshua Evans and his team, where they also made novel misos also on different substrates and with different fermentation conditions. And they could also say that both the substrates and fermentation conditions have a huge impact, not only on the flavor outcome, but also on the microbial communities that are formed. And then we paired up with Joshua and his team, and we wanted to expand basically this concept, and thought, okay, maybe it's also the environment, because people are talking about the terroir of a specific place, and that might also have an impact on the microbial communities that are formed and the resulting flavors. And Joshua was working together with Maggie Koblenz, who was part of the MIT Space Exploration Initiative. So we got really lucky that we were able to explore really extreme conditions. So we had misos made with the same recipe and in the same, as the same batch, but then they were shipped to 3 different locations, 2 on Earth, 1 in Cambridge, 1 in Copenhagen, and 1 at the International Space Station. Then it was actually the first time that that something actually fermented in space. So the first question was, is it even possible to ferment in space? And then we also wanted to investigate the impact of the environment and the associated fermentation conditions on the microbial communities and flavors. And we had a sensing box that actually measured the conditions, and we had that also on one of the samples on Earth. And here you can see that also the fermentation conditions changed dependent on the environment, which might have had then also an effect. And overall, if you look at the microbial communities that formed, we see that overall they look somewhat comparable and similar across the different locations. But then there are also small differences, which might be attributed to the fermentation conditions as well as the location. We also assessed the mutation rate of the Aspergillus oryzae, which was slightly elevated at the space station, which means that maybe the radiation, which is also an environmental condition, also had an impact there on the genetics. Similarly, we assessed the flavors and aromas and did also sensory analysis as well as metabolomics. And we do see that overall there is similarities, which means that fermentation in space is actually possible, but there are also some differences which are then again attributed to both the environment and then the associated differences in the fermentation conditions. And here you can see Joshua Evans and Maggie Koblenz, who led the study, actually while drinking a miso soup that was made from the leftover miso from this project that was then made into a dinner by a chef called Kim Weindorp that I also mentioned earlier, to basically value the mesas that were created in this study and to make sure that they don't just sit in a fridge for the rest of their lives. But to summarize this section, basically biodiversity in fermented foods is linked to flavor and can be controlled through specific conditions, especially the substrate influence. The substrate influences the food fermentation microbiome a lot and the resulting flavors, but also the fermentation conditions have a huge impact, as well as the genetics of the starter culture and maybe also the fermentation environment. And with that, I want to go back a bit to some challenges of the food system, which are also linked to diversity. So if we think about all the food that we are eating or not eating, it becomes apparent that there is like more than 30,000 edible plant species, but we are only cultivating 30 plant species. And interestingly enough, more than 50% of the carbon atoms in our bodies are actually coming from corn. So we're not eating that diverse. Similarly for animals, even though I think it's less important given that I think we should move away from animals, but there's probably way more species of animals that are potentially edible than plants because very few are toxic. But then we are also farming only very few animals. And the same holds true for microorganisms. So there's probably thousands of potentially beneficial food fermentation organisms that are probably also in traditional or spontaneously fermenting foods. And there's only very few starter cultures that are commonly used, and oftentimes they are even replaced by enzymes that do the job, that are isolated from the microbes, or fermented foods are replaced by non-fermented foods where we no longer rely on the fermentation process to make them, which means that we have even less exposure or microbes in our diets. And I think this is due to a matter of scale, actually, and the type of production that we do. So basically, initially we would forage and then we would be able to access this huge biodiversity, and we would otherwise hunt and gather, which again leaves us to a lot of diversity in our food choices, and we would traditionally spontaneously ferment foods, which also contain a lot of microbes. But then that meant that everyone was actually involved in making food for themselves. Then we started introducing agriculture, and we narrowed the number of species that we would actually cultivate. But that also meant that only 70 to 90% of the population had to be doing agriculture, and there were other people who could do other tasks. Then we started in the Green Revolution to start intense agriculture and intense animal farming, and also to industrialize food production even more. And I think that a lot of those technologies are then built around the food, but then they're all very specific foods, which means that the technologies only work for that specific crop or that specific way of growing or producing it, which leaves a lot of the other crops for us that they are not suitable, or other crops cannot be grown, for example, if they can't be stored and transported. So there's very a lot of selection criteria why we had to narrow down the number of species that we are currently eating. We also want to control the production and have kind of reliable outcomes. And flavor profiles, for example, and that is another motivation why we want to kind of try to control and have reproducible production systems. At the same time, that means that today only 4 to 30% of the population are involved in food production. But then this is actually also connected to the resilience of our food system. So relying on very few species in a highly technological system requires a lot more inputs. So we need to use fertilizers and pesticides in order to grow our crops, or we need very sterile production processes with dedicated equipment and trained staff to produce fermented foods, like you can see yogurt being fermented here. But that also means that the system is very vulnerable to new pests or other challenges. For example, we only produced one clone of banana, and then there was a pathogen wiping out all the banana plants, and then we lost this whole crop or this whole strain of bananas forever, and now we're growing a different type of banana, but again in monocultures and requiring a lot of input and control. And similarly, for example, I found a story about the Roman Empire where they used to use garum for almost all the foods that they are producing. And then that was lost because when the Roman Empire fell, the factories that were producing it didn't have the infrastructure for transporting ingredients and for selling them. And the knowledge had been only maintained in the factories and not at household level. And then garum just disappeared from the Roman diets. So again, I think we should be inspired by nature and think of the circularity, but also the diversity in the food system. Here you can see industrial yogurt making, which requires specialized equipment and infrastructure, as I mentioned, and that is usually made from 2 individual isolated species of lactic acid bacteria. And the process needs to be kept sterile because otherwise phages or other contaminants would kind of take over. Traditionally, yogurt was produced at kitchen scale and was needing less equipment and input materials and could ferment spontaneously. And those traditional yogurts contain more than 75 different species, and including different types of bacteria and yeasts. And if you use industrial yogurt to make new yogurt from milk, usually this works only for a few generations and at least at kitchen scale. While traditional yogurt can be used for backslopping, which highlights that the diverse communities of microbes are more robust and better also under less controlled conditions. And then dairying was actually started in the area, or at least the first archaeological evidence that we have of yogurt fermentation comes from Turkey and Bulgaria. And we have collaborated with Sevgi Mutlu, who grew up in Bulgaria with Turkish heritage. And in her village, the ancient tradition of starting yogurt with ants was still practiced, and she reported or is actually researching this. And me and a team of interdisciplinary researchers got really interested in that because we wanted to know whether that can lead us to the origin of some food fermentation microorganisms, but also to understand the natural biodiversity in fermented foods and also diversity of creating foods in general and production processes. So in a team composing, for example, or in a team that was led by Veronica Sinot, whom you can see here, who was accompanied by David Silber. They went on a field trip together with Sevgi Mutlu into her hometown in Bulgaria, where they tried to re-establish this traditional yogurt fermentation using ants, where they buried actually the yogurt in a ant mound, which serves not only the purpose that ants enter the milk and form yogurt, but also to hold the temperature. And then they took samples and studied that in more detail. And at the same time, we also recreated the ant yogurt in the lab. And here you can see Veronica Ramos-Viana doing ant yogurt under otherwise sterile conditions to really test what are the contributions of the ants in order to make yogurt. And we found that ants actually contribute microorganisms, and that those microorganisms form very stable and controlled microbial communities, which is good for fermented food. And a lot of those microbes actually come from or are lactic acid bacteria, and they proliferate in the yogurt, which is also quite exciting. We also tested the acids that are introduced through the ant, and we found that the ant and the microbes associated to the ants increased the acidity in the yogurts, and also contribute proteases, which can also influence the texture of the resulting yogurts. So the whole ant holobiont basically contributes to the yogurt making. And to come back to the initial, like basically questions that we asked, we actually found that a food fermentation microorganism, namely Fructolactobacillus sanfrancensis, is carried from the ants to the yogurt. And this microbe is actually associated with sourdough. So it's very interesting to think of the ant as an ecological reservoir of really food-relevant microorganisms. And inspired by that potential, we partnered up again with the Alchemist restaurant, who did like an ice cream in the form of an ant from the ant yogurt and also other really interesting stuff to really highlight the potential of food innovation from like bridging basically this traditional approaches to modern gastronomy. And they also tested the potential for plant-based yogurts to be produced using the same traditional approach, which was very promising and I think really exciting. And while this innovation potential initially drew me into the project, and I still think that it is really exciting to identify new ecological reservoirs of microbial biodiversity for food innovation, I think I have to admit that I also learned something else in this project, and something that seems in the end much more important to me. So Veronica told me after her field trip in Bulgaria that the people in the village could taste which cow gave her milk for the yogurt and which family had prepared the yogurt. And this aspect really stuck to me because first of all, I think it is really beautiful to think that we can actually taste the contributions of different human and non-human individuals who contributed to making a food, including the animals and plants that went into it, but also the microbes, as I presented earlier. If they are communicating, we can actually listen into those communications and different contributions with our senses. And by tuning in and listening to our senses, I think we can then actually, through food, connect not only with our fellow humans, but also other fellow species on Earth. And fittingly, the word for starter culture in Bulgarian is called Maya, and that comes from the interconnectivity of the web of life. And I think that is really illustrating that point. And I think feeling yourself as a part of the web of life has many positive aspects. So you understand yourself as part of a community that nurtures you and supports you, which might be quite comforting. But it also makes you realize your dependencies and interactions with other individuals. And I think that might actually provoke feelings of gratitude and compassion and care, at least in myself. And I think that these feelings can also be transformed in taking responsibility and care for life on Earth. And I believe in the end, if everyone would feel in this way, we would be much closer to finding solutions for our social and sustainability challenges. And finally, I want to bring that back to the diversity on the level of food production methods. So we usually talk about scaling up and industrialized processes to generate impact and reach many people. But I hope I've convinced you that there is also value for everyone to take part in foraging and food fermentations themselves to diversify not only the food that we are consuming, which is probably also very healthy, but also to connect to life in a meaningful and actually deliciously rewarding way. And in addition to scaling up food production solutions, we can consider scaling in different dimensions. For example, we can scale out food production across many individuals who make their own food and kitchen scale. And this has likely the advantage of requiring less equipment and infrastructure and training, and is therefore a very inclusive and democratizing approach. Also, it makes the food system more resilient if many individuals and systems are providing redundancy. And before I conclude, I want to quickly say that scale out also doesn't mean that everyone in the world should make ant yogurt now, because that evolved in a very specific cultural and geographical context, but also because ants carry actually a pathogen, and we had to take some special care in order to not infect anyone with this. So just, yeah, stay safe, one could say. But there's many different ways to contribute and actually engage in the food system. So you can go foraging, you can grow your own veggies in the garden. So you can see here some harvest of my spring salad here, and you can grow many different varieties that you don't find in the supermarkets. You can also ferment your own food, and all of this will work best for species and food types and traditions that are somewhat native or pre-adapted to the environment where you live. And with that, I want to finish my talk. So the diversity of food production methods and scales can allow us to increase the biodiversity in our food system and to improve the resilience of the food system and to connect people back to the natural world. And being part of scale out of food fermentation as well as farming allows you to connect to the natural world, to take responsibility, to take care of the life around you, to taste the beauty of life and multi-species interconnectivity. And yeah, basically more diversity and interspecies connectivity in our food system will help to create more sustainable food systems. It can improve the resilience of the food system. And if we scale out in addition to scaling up, we might also develop solutions that can be accessible around the entire world without specialized equipment, infrastructure, and training to allow for a more equitable and just food system to develop. And with that, I want to thank everyone who contributed to this talk and the Microbial Foods Lab and all our collaborators, as well as all the funding that went into this project or the different projects. And of course, you for your patience and your attention. Thank you.
Thank you so much, Leonie. That was fantastic. Many comments throughout that are just thanking you for a really great presentation and for also giving us more than just technical research itself, helping us kind of zoom in and out at the societal consequences of microbial diversity. So very appreciated for the many years you spent doing this research and also the time you've taken to share it with the scientific community. For those of you in the audience who have not yet gotten the chance to do so, please drop any questions that you have for Dr. Jahn in the Q&A section of your Zoom bar. We'll shift over into Q&A now, and Leonie, feel free to stop sharing your screen. And we'll go ahead and shift into some of the questions that audience members have for you. So one of the first questions folks have is around your thoughts on whether we might be able to achieve economies of scale with these technologies so that they might be cost competitive. You had your scaling section towards the end of your presentation where you talked about not just scaling up, but also scaling out. But I'd love to hear a little bit more on your thoughts on what the path might look like for really scaling microbial production.
Yes, I think it's a very valid question and something I think a lot about actually in my daily life. And I think that we probably need, again, redundancy in our approaches. So scaling out will not solve all our problems. I think it's a very beautiful approach, but I think a lot of people don't want to engage in it or spend time on it or don't have the ability to do so. And I think we need more convenient and traditional, not traditional, conventional maybe approaches as well to complement that. And I know that for fermented foods, the scale up is a big issue, one could say. So there's a lot of products being developed at kitchen scale that really work well with the craftsmanship and basically the care that an individual can put into it. But then if you try to scale that up, that becomes very challenging. But I do think that the field is moving pretty fast there and that we will actually come up with solutions to do that. And at least if we talk about tempeh, for example, which is a nice example of a solid state fermentation with the filamentous fungi that has been successfully scaled already many years ago in Indonesia, where it was first developed or where it's originating from. And I think that highlights that it is possible. But I also know that every ferment is different, every microbe is different, and it will need some research in order to get there.
Thanks so much. This is a related question. We think about scale, we think about cost, and many folks in the community come to the alternative protein space because of their curiosity and deep interest in resolving global food insecurity. So when we're thinking about some lower income contexts, some developing economies where access to high quality proteins might be more limited, I think fermentation comes into the conversation a whole lot. So when we think about applying everything that you've just shared to a developing economy context, Might there be a starting point, like a substrate or a starter culture that you would recommend starting out with to look at the application of this technology to a very large scale, very low
Mm.
cost type of environment?
Mm. I mean, I think this is also very relevant and important to consider. And I think it's interesting because I feel like in Europe and the US, we have actually lost a lot about like a lot of our traditional heritage in terms of food fermentation. And now we're drawing actually from a lot of other countries around the world. And I think the more food insecure a country, actually, the higher is the rate of fermentation and traditional fermented foods that you can find there. Like in Sudan, for example, I think there is like 70% of all the food that is produced there is actually fermented. So I think actually that especially these places in the world have excellent heritage and actually cultural knowledge and are deeply rooted into producing fermented foods and actually like unlocking, like for example, poisonous foods through fermentation and like basically yeah, making them safe to consume or to improve the nutritional qualities of foods or the palatability of foods through fermentation. And I think it's maybe more a matter of finding that confidence in their own cultural heritage that is actually already there, and a lot of the solutions are already there. And then maybe innovating with that in terms of scale, if need to be, or at least make sure that the cultural heritage isn't lost and is kind of maintained in the population, that people can continue to create their food in this way.
Thank you so much. There's a question here around where you might actually find suitable fungal and microbial strains. So, can you speak a bit about the natural environments that you might find strains that are most suitable for domestication?
Yes. So for fungi, at least, if we look into traditional spontaneous fermentation where we didn't use starter cultures, we actually find, for example, for tempeh, the origin is that the soybeans were wrapped into banana leaves, which then also carried the spores of the fungus that then spontaneously started fermenting. And now we're using starter cultures because of the food safety aspect, I think. But I also think that traditionally there were potentially safety measures in place that maybe have been lost, but are still kind of, yeah, in the wisdom of how the fermentation was done exactly in terms of salt concentrations or treatments, pre-treatments or steaming or those kind of things. So I think for fungal fermentations, there's other examples as well where the fungi come from leaves and different plants. So I think that could be a good starting point. For other dairy fermentations, there's a whole suite of different substrates where we might find microorganisms that actually started off yogurt or cheese fermentation. And similarly, this may be a bit different for plant-based ferments, where then usually the microorganisms are already on and in the plants and can actually oftentimes ferment spontaneously. So I think like looking into those traditions can at least give a starting point for finding those ecological reservoirs of food fermentation microorganisms. But at the same time, the example of corn also highlights that you can also look into completely different species and really try to find new ways of innovating with food. So I think both things could be explored in parallel to really cover this whole space of potential.
Thanks again. You spoke a lot about the use of diverse microbial communities in maybe helping enhance flavor in alternative protein products. Can you talk a little bit more about how microbial diversity might help with enhancing texture in alt protein products?
Yes, of course. I think, like, again, we're trying oftentimes to add enzymes or specific molecules like methylcellulose, for example, into fermented or not, maybe also just plant-based foods in order to generate texture. And I think this could also be a very interesting angle to look at microbes that are producing cellulose, for example, or microbes that are producing enzymes that cross-link proteins, and add them to fermented foods, also to fungal fermented foods, to see whether this is a way to potentially also improve texture.
Thank you. Can you speak a bit about the regulatory considerations? I think it's great that you are both an academic researcher and have kind of embarked on a startup journey. So just there's a question here from an attendee who's curious whether you've run into any regulatory challenges selling products made of Pleurotus austriatus mycelium in the EU or other microbial strains experimented with.
Yes, so yeah, in the company we haven't faced that problem because yeah, we're relying on approved ingredients and strains. With the Pleurotus austriatus, yeah, actually the dish is actually discontinued on the menu of The Alchemist because of that reason. Yes, so I think there is some issues there. I think in general the food scene in Copenhagen and the restaurants have explored of novel ingredients, and that is also kind of at the core of what they are doing. So I think that is yeah, there's a balance there of how to approach that. On the other hand, a restaurant is also different to a company, and also again different to what you do at home. So there's also a whole range of different like rigidity in terms of the regulation that needs to be applied. And for a company, I would certainly only, at least for now, look into approved strains and consortia. Potentially, I mean, one could also embark, of course, on novel food approval. It just takes more time and funding, I guess, to go through that route. And I think there is a lot of potential there as well. It just, yeah, it's probably not the first product you would want to launch, I guess.
Yeah, I have read, in just conversing with others across the ecosystem, that a pretty safe governing philosophy is that if you're exploring new microbial strains, you really want to start very, very early on by understanding the regulatory pathway. If it isn't yet approved, at least understand whether there is a pathway for this particular strain to meet the specifications required by various governments around the world. Otherwise, you will hit that roadblock at some point. It is better to learn it sooner rather than later. Awesome. One attendee is curious if you're able to share, totally understand if not, but if you're able to share what strain you use to produce your Matter burger. I think sensing that's a hard
I'm
no.
unfortunately not allowed to talk about that.
That makes a lot of sense. Thank you. There's a question here around making flavors for dairy alternatives. Hoping you can speak to that a little bit more. How might someone kind of tweak this entire body of work around using mushroom mycelium and the diversity within that microbial ecosystem for dairy alternative flavors?
Well, I guess there's many different things that would come to mind how to approach that. I mean, one can ferment plant-based substrates with filamentous fungi similar to like blue cheese or camembert, basically, in order to, yeah, also introduce flavors and textures. There's also, I think, Formor is a company from Germany that uses koji biomass as an ingredient for dairy products or dairy alternative products. So I guess also fungal biomass in itself could be a valuable source of protein that can then be formed into new products. Yeah, and I guess also, I mean, in principle, you could also envision all sorts of additional ways of where you extract maybe specific compounds or flavors or enzymes that could then be added or be able to treat certain ingredients to get to a specific flavor or texture profile. So I think there's, yeah, a lot of room actually to explore that.
Thank you so much, Leonie. I think that's the end of all the audience questions for today's seminar. I would really love to ask you a final question before we wrap up, which is just around what you would like to impart on the global scientific community. Any last words you'd like to leave the scientific community with today as we wrap up our seminar?
Yeah, I guess for me it's really important to collaborate in general and to break down silos and to just, yeah, collaborate across disciplines and across sectors. And I think that that is also really important for moving forward for the community in general, especially if we work on food, because food is really holistic and tied to a lot of, yeah, cultural and emotional aspects, but then again also to so many different scientific disciplines. So I think I can only encourage everyone to do the same and try to speak to many different people and get many different perspectives on this problem.
Yeah, it absolutely is an interdisciplinary challenge and a beast of a scientific undertaking. So I think that's a really great set of parting words. Thank you so much again, Leonie, for sharing this research with our community. Thank you to all of you for attending today. Do continue to stay in the loop, and I hope I see all of you at our next Science of Alt Protein seminar next month. All right. Thanks so much, everyone. Thanks, Leonie. Bye.
Thank you. Bye.